Hello, my name is Tyler Figliuzzi, and I did my research on the separation of Ig proteins from cow milk and colostrum for use as supplements or food additives. Dairy products are one of the most abundant sources for bioactive proteins, like bovine milk and colostrum. Separating and isolating specific bioactive milk and colostrum components will allow nutritionists to use them as supplements for patients with weak immune systems, like seniors and infants, while benefiting stakeholders like cow farmers. Important milk proteins include casein and whey, but this review will focus on Ig. The goal of this paper is to show the opportunity that Ig protein supplements have to help the human immune system. The intended end users will be those with a weak immune system, generally infants and seniors. To accomplish this, general information will be discussed about Ig along with the bioseparation technique. Ig are already found in the human immune system as antibodies. Antibodies attach to foreign substances in the body and allow the immune system to destroy them. Ig accounts for about 1 to 2% of proteins in milk and about 70 to 80% of proteins in colostrum. They are split into two main classes based on their functional properties, which include IgG and IgA. By using Ig supplements and additives, it will enhance the human immune system and help fight off illness. IgG is the smallest antibody and are very important in fighting harmful bacteria and viruses. As it can be seen in the figure, IgGs are composed of two heavy chains and two light chains linked by four disulfide bonds that form a Y shape. Each molecule contains a fragment antigen binding domain and a fragment crystallization domain. When IgG are called into action, the CDRs in the FAD domain bind to the foreign body. Receptors binding to the Fc domain are called Fc gamma receptors. Once the immune cells bind to the receptors, the Fc domain mediates the effector functions, like the killing of the foreign body. By having more IgG proteins available through supplements or food additives, those with weaker immune systems will have a stronger chance of fighting off any sickness involving harmful bacteria and viruses. IgA antibodies help protect the body's surfaces that are exposed to outside substances. Those are two subclasses of IgA, which include IgA1 and IgA2. The structure of IgA is very similar to IgG. As seen in the figure, the heavy chains are folded into four glob chains, while the light chains are folded into two glob chains. For IgA proteins, the most active receptor is the Fc alpha Ri receptor. Once the protein binds to the foreign body, the receptors bind to different immune molecules through the interaction between the receptors and the C alpha 2 and 3 chains, which induces the elimination process through different mechanisms. For those with weaker immune systems, having the ability to consume IgA proteins, they'll have a higher chance of defending against illness caused by outside foreign bodies. The bioseparation of Ig proteins from bovine milk and colostrum is required for the downstream process of creating food additives and supplements. There are many bioseparation techniques, but this review will focus on chromatography. This technique is based on electrostatic interactions between charged proteins in a matrix. The environment pH that creates no protein charge is called its isoelectric point. IP can be calculated based on the primary sequence of the protein, and the buffer pH will determine the net charge of the protein of interest. As seen in the figure, if the protein is positive charged, a negative charge anion exchange resin should be chosen. The opposite will be true for a negative charged protein. This technique separates macromolecules like proteins based on their size. In the column, the solution with the protein runs through a porous gel. As seen in the figure, the molecules that are larger than the pores do not permeate and pass through faster, while the smaller molecules diffuse into the gel and take longer to travel through the column. Bovine milk and colostrum contain many bioactive proteins, including Ig. Ig proteins act as natural antibodies by attaching to any foreign substance in the body and allows the immune system to destroy it. Taking Ig protein supplements or additives will strengthen the human immune system and help the body defend against illness, especially for infants and seniors. Due to proteins' physical characteristics, ion exchange or gel permeation chromatography will be as successful in separating them, but will have limitations like protein denaturation through temperature. To keep the process efficient, it is recommended to use ion exchange chromatography and to keep the environment temperature below 65 degrees Celsius throughout the entire process. Due to the antibody properties of Ig, and by keeping the downstream process as efficient as possible, the Ig supplements can help support the health and nutrition of infants and seniors and society as a whole.